All right, so today we're going to do reproduction and inheritance. Uh, this chapter is very big and long, so there's a lot of stuff you need to memorize in this chapter. So I looked at the 2023 syllabus for the IMAT and I compared it to the old one, and there wasn't much of difference in both of these syllabuses. So everything is the same except that now the the what do you call that, psychology is in the psychology part is now in bioenergetics. That's the only difference. Other than that, everything is the same. There's no reduction or increase in the syllabus. So yeah, so let's start with the Mendelian inheritance terms. So for the exam you have to know some definitions. It's very basic, so let's start with that. Genotype is the allele passed by an organism. A genotype is the allele possessed by an organism. So, for example, capital A, small a, this is called an allele, and this is the genotype of the organism. Similarly, a phenotype is the observable characteristics of an organism, and phenotype is basically what you can observe on that organism. For example, the animal can be long, it can be tall, it can have different color of hairs. All of these are the phenotypes. And phenotypes can be influenced by genotype and environment, whereas genotype can only be influenced by mutations and the variation in genetics. So like independent assortment that happened in meiosis, that's where genotype can be changed, and as well as mutations. So a genotype, right? What is a gene? A gene is a specific DNA sequence that codes for a polypeptide, and it's represented by alphabets. So capital B is usually called the dominant allele, and the small letter refers to the recessive allele. Yeah. So what is the allele? It's a form of gene, and it is a result of mutation. The dominant allele is the capital letter. As I said, the recessive allele is the small letter, and the dominant allele will always be expressed in the phenotype. So the dominant allele will always be the superior, which gives you the phenotype. We'll discuss more about this, but for now, just know this. And similarly, a diploid cell is two sets of chromosomes, so two alleles. And what is homozygous? Homozygous is when you have two identical alleles of a gene. So for example, you have two dominant alleles. This is homozygous. But what is a heterozygous? A heterozygous is two different alleles of a gene. For example, I have big B and small b. This is heterozygous because I have dominant as well as recessive. In homozygous, you can only have either recessive or either completely dominant. Heterozygous, you can have a mixture. So one dominant, one recessive, that's called heterozygous. Yeah. So moving on to dominant alleles. Dominant allele is expressed in phenotype even when only one copy of allele exists. So for example, I have this is heterozygous, right? And when I have this capital B, this will be expressed as the phenotype. And it's expressed in both homozygotes and heterozygotes. What do I mean by this? Capital B, this is homozygous. And this right here is heterozygous. In both these cases, in both of these, my phenotype would be determined by the dominant allele, which is this right here. So the phenotype is given by the dominant allele in both the homozygous and the heterozygous. Whereas recessive alleles is expressed in phenotype only when you have two copies of that allele present. and it's expressed only in a homozygote for that recessive allele. What do I mean by this? Similarly, if I had a homozygous, this is homozygous for recessive alleles because I have two similar recessive alleles, so this is homozygous. And heterozygous is when I have one dominant and one recessive allele. That's called heterozygous. And recessive alleles can only be expressed. So, for example, I had capital B. For capital B refers to long hair. And small b, this is the recessive allele, refers to short hair. In this case, when I have homozygous for recessive alleles, when I have both the alleles as recessive, then only can I express the recessive allele for the phenotype. So in this case, this allele will express the short hair phenotype because I have two recessive alleles. When I have a heterozygous, right, when I have a dominant and a recessive allele, the dominant allele would express the phenotype. So in this case, my long hair would be expressed. So remember this rule that whenever you have a dominant allele, that would be expressed. And yeah, so there is some examples here. Let's focus on that. For example, I have homozygous dominant. So B refers to brown hair. Capital B refers to brown hair. And small b refers to black hair. When I have a homozygous for dominant alleles, when I have two capital Bs, then my brown hair would be expressed. When I have a heterozygous for dominant alleles, meaning one capital B and one small b, then my capital B would be expressed. So I will have brown hair. If I have a homozygous recessive allele, meaning I have two small b's, then my black hair would be expressed. All right. Yeah. So this was very basic stuff. Now, you also need to know how to identify and draw dihybrid cross and monohybrid cross. For IMAT, you only need to know hybrids because that's for your exam. But for A levels, you need to know dihybrid. So you can ignore this. We'll only talk about monohybrids. In monohybrids, right, you have two alleles being expressed. So this is one allele and this is another allele. So one capital and one small, one capital, one small, one capital, two capitals or two smalls. These are the three combinations that you can find for the exam. And it always starts off, you won't be asked to draw all of this parent genotype. What you need to know is how to identify ratios. So for example, I saw a question in the exam. They told me that, hey, the parents, the male parent has brown hair, right? And brown hair is capital B. So I'll say brown hair is capital B. And for small hair, for black hair, it would be small b. So this would refer to black hair. Now, this question came in the IMAT. So they told me that, hey, male has brown hair and it's homozygous. Whereas the female parent, the female is black hair and it's homozygous as well. And they tell me, the question says, what is the ratio or the percentage? They can also ask you the percentage that my offsprings will have black hair. So this is the question. They're asking me how many children, or what's the percentage of my offsprings of this two male and female when they cross, how many offsprings will have black hair? This is the question. Someone is saying 0%. We can figure that out. First of all, they told me the male is homozygous and he has brown hair. When they say it's brown hair and it's homozygous, I know that I'll use the capital B. So two capital B's, because they say it's homozygous. So homozygous means two similar alleles. Brown because it's capital. And homozygous for recessive here. That means for black, I'll have small b's, and this is homozygous. When they cross, uh, how do I draw the cross? How would I know the genes for my offsprings? Well, I would write these b's separately. So this is for the male, and then I'll write the female right here at the top. So small b and small b. So this is how I draw the the diagram for finding out my offspring genetics. So then I do this: capital B, small b, capital B, small b, capital B, and then move on to the next one, small b, like this. And similarly, capital B, small b, capital B, and small b. So all of these are heterozygotes. All my 4 offsprings are heterozygotes, and they have the dominant alleles. So when something has the dominant allele, that would be expressed. So I have brown hair. All my offsprings would have brown hair. Therefore, the percentage of my offsprings having black hair, uh, what is the percentage that the kids would have black hair? It's 0%. So that's the answer. This is how you find genetics for offsprings. Yeah. So this is how you find genetics for offsprings. And so remember, there is something called purebred as well. Uh, Purebred is when you cross the same purebred organisms, and they will always produce offsprings with the same genotype. So homozygous individuals. So what is purebred? The same alleles. For example, if the parent had BB, capital B, small b, then the offspring would also have the same allele. So this is known as purebred. And yeah, so um, yeah, so moving on to Mendelian laws. There are 2 laws for Mendelian genetics you need to know. It's called the law of segregation. Law of segregation states that during gamete formation, when gametes are formed in meiosis, the allele for each gene segregates from each other during anaphase, so that each gamete carries only one allele for each gene. So when the chromosome, they separate, right, and they move to the opposite ends, right? This happens at the anaphase 1. This is segregation. So this is called the law of segregation. And the law of independent assortment is when each pair of chromosome lines up independently of one another on the equator during metaphase 1. So genes of different traits can segregate independently during formation of gametes. Law of independent assortment states that during metaphase 1, my chromosomes would line up independently of one another. This is basically law of independent assortment. There is another law of dominance, but this is very basic. You already know this, that the alleles of the dominant alleles are always expressed compared to the recessive alleles. So this is not that important, but you have to know these 2 laws. So moving on to the different types of combination of monohybrids I can have. So there are 5 different ways genes can be expressed. When you have sex chromosomes involved, meaning X and Y chromosomes, these X chromosomes comes from the female, and Y chromosome comes from the male. And when you have sex chromosomes involved, this type of genes are called sex-linked. Uh, and when something is not exactly recessive or dominant, it's called codominance. You don't need to know this. This is not in your syllabus. For the syllabus, you only need to know sex-linked, and the other one is autosomal linkage. That's what's written in your IMAT syllabus. So what is autosomal linkage? When genes are present on the same chromosomes, that's called autosomal linkage. For example, I have a chromosome and the alleles are present on the same chromosome. That's called autosomal linkage. So remember, autosomal linkage only happens in somatic cells. We learned this further. somal basically refers to somatic cells. So let's start with sex-linked genes first. So in sex linkage, the allele or gene is carried on the sex chromosome, and it's usually on the X chromosome. The Y chromosome, remember that the Y chromosome will not carry uh, any alleles because the Y chromosome is very short. When you have X chromosome, that chromosome will carry the allele. So yeah, so the X chromosome carries the alleles, and you can have two types of sex-linked inheritance. The first one is X-linked recessive, and the second one is X-linked dominant. So let me explain this through a diagram, because in most of the questions I've seen for the IMAT as well as the BMAT, I found that they usually give you inherited diseases, inherited genes that come from X and Y chromosomes. So they'll usually ask you sex-linked questions. So how do you do this? And I'll explain this through a question they asked for the IMAT. They told me that, hey, they told me that there is a male and a female. Females obviously have XX chromosomes. Males always have XY chromosomes. Remember this. Males have XY, females have XX chromosomes. And they tell me that the female carries a disease, and the alleles for the disease is present on the X chromosome only, and it will only be present on the X chromosomes. Remember that alleles on X will be present on X chromosome, not the Y chromosome. So Y chromosome doesn't carry anything. And they tell me that the disease has two alleles. One is, let's say, A, capital A refers to dominant. That means if this is expressed, then the disease would be present. And small a refers to recessive. So if I have the dominant allele, then I will carry the disease. If I have two recessive alleles, then I won't carry the disease. So this can also be reversed. And they can ask you that, they can say that, hey, A refers to recessive and small a means dominant. What do I mean by this? This means that if I have two recessive alleles, then only will the disease be expressed. And if I have a dominant allele present, then that's the allele for healthy cells, and then I will not have the disease. The disease would not be present. So if I have two small recessive alleles, then I will get the disease. If I have one dominant allele, then I will not have the disease. So in this case, what the question told me is that the dominant allele, if it's present, then I'll have the disease. If the small allele is present, if two small alleles are present, then I would have two recessive alleles, then the disease would not be expressed. So they tell me that the female, right, this female carries the X chromosomes, and they tell me that it's A and small a. So one chromosome, X chromosome, has A, the second one has small a. Whereas for the male, the X chromosome has A only. So remember, female contains the dominant allele. Dominant means that I'll carry the disease. So female is a carrier. When some organism carries the disease, we call it a carrier. The male part only has the recessive allele. And when I have a recessive allele, when I have only one recessive allele, then the disease will not be expressed. Because I can't have two recessive or two dominant alleles in a male. I can only have one allele, either capital A or small a. So in this case, they told me that the male has small a only, and this means that it's recessive, so it would not be a carrier. It would not carry the disease. So they tell me that they interbreed, and they tell me what is the probability or the ratio that the offspring will carry the disease. What's the percentage? So how do I do this? Again, I make my diagram for offsprings. I write the male first, XA, right, the recessive allele, then Y, and then female, I write X capital A, dominant A, and then small a. And then I simply do the simple maths. So X small a, X capital A, and here XA, XA. You have to usually write this as first, but that doesn't matter. And then Y and XA, and Y and XA small. So Y and XA small. So how many offsprings carry the dominant allele? 1, 2, and so 2 offsprings have the dominant allele. This means that in these 2 offsprings, the disease would be present. So what is the probability that my offspring will have the disease? It's 50%, because out of the 4 offsprings, 2 will carry the disease. So it's 50%. And sometimes they confuse you with the question, and they, instead of asking you, I've seen a lot of this question, instead of asking how many offsprings will carry the disease, they'll ask how many male offsprings carry the disease. So when they refer the gender, let's say they asked you what's the probability that the male will carry the disease, then you will look at the males only. So the males would have the Y gene. So in this case, this right here and this right here. You can see that only one of the males have the dominant allele, right? The other male has this recessive allele. That means this out of the males, only 50% of the males will carry the disease. There is a 50% chance that the male will have the disease. So yeah, if they asked how many females will have the disease, then you look at the females only. So when 2X are present, it refers to females. And you can see that only one female has the dominant allele. So you'll say that 50% of female will carry the disease. Is this clear till now? So that we can move on to autosomal linkage. Is this clear? All right. So this is how you do sex-linked diseases. So yeah, and for the exam, you also have to know the inherited diseases. For the exam, you have to know all of these 5 diseases, and specifically, you have to know the autosomal ones. So this right here, autosomal ones, as well as X-linked recessive. So if you have sickle cell anemia, that would be caused by codominance. You only have to know sickle cell anemia is caused by codominance. And what happens due to sickle cell anemia? Your red blood cells would be sickle shaped. That's, as far as I know, you only have to know this much, because this is for codominance, and codominance is not in your syllabus. For albinism, you have to know a lot, because it comes under the autosomal recessive category. So albinism is when you have loss of the melanin. So when there is no melanin, you will have albinism. We learn each disease one by one. So yeah, let's start with albinism. So albinism is caused by autosomal recessive mutation. Remember what causes these types of diseases. For let me go back. For albinism, it's caused by autosomal recessive. For hemophilia, it's caused by X-linked recessive. So this is a sex-linked gene. So if they asked you in the exam what type of inherited disease is caused by hemophilia, you'll say it's a sex-linked disease. Similarly, Huntington's disease is caused by autosomal dominant, and cystic fibrosis is caused by autosomal recessive genes. And you also have to know all of how it's caused and why it's caused. So starting with albinism, it's caused by autosomal recessive mutation. Uh, it's when melanin is missing from your eyes, skin, and hair. And there is a mutation in the TYR gene coding for the tyrosine enzyme. And so this tyrosine enzyme, right? This tyrosine enzyme isn't sickle cell autorecessive, isn't uh it's autorecessive. Wait, sickle cell anemia is autosomal recessive, not codominant. Uh, as far as I know, it's caused by codominance. Uh, so the all right, if we want, we can learn sickle cell anemia, but let me check on Google if it's codominant. The altered form of hemoglobin causes sickle cell anemia. It is inherited as a codominant trait. So yeah, Google is also saying it's caused by codominance. Yeah, it's caused by codominance. Hmm. You know what? Let's study sickle cell anemia as well. What will happen if we study it? We can study it at the end if we get time. But for now, let's focus on these diseases, which we know that are important. So again, tyrosine enzyme, there is a mutation in the tyrosine enzyme. Tyrosine enzyme converts tyrosine into something called DOPA, DOPA. And this DOPA is needed for melanin production. Melanin production by what? How is melanin produced? Melanin is produced by melanocytes. Remember, so in a summary, mutation happens in the tyrosine enzyme. Tyrosine enzyme is needed to convert DOPA into melanin. And if you don't have this tyrosine enzyme, then you won't have this conversion from DOPA to melanin. So no melanin is produced by the melanocytes. So when someone has albinism, then he'll be the color, there would be no color. So he'll be white. So his eyes, his skin would be white. And you have to know the symptoms, how can you figure it out? So moving on to hemophilia. Hemophilia is a sex-linked disease. It happens because there is a gene that codes for blood clotting protein called factor VII. And this gene right here is carried on the X chromosome. So there is a mutation for this protein, this gene that codes for the protein. And this will cause the X-linked recessive mutation, and you'll have hemophilia. So remember, hemophilia is sex-linked, and it's caused by X-linked recessive mutation. So there is a mutation on the gene present on the X chromosome, and this will cause the factor VII. This is the protein. This will not be produced when you have the mutation. And the symptoms for hemophilia is you will have excessive bleeding because the platelets would not be able to clot the blood. And similarly, you will have bleeding internally as well. So this is what you need to know for hemophilia. For Huntington's disease, it's a neurological disorder. It is caused by an autosomal dominant allele. So if only one allele of that gene is present, then you'll have the disease. And the mutation happens at the HTT gene, HTT gene. You also need to know the chromosome number. It's chromosome 4. And what are the symptoms? You'll have involuntary movement. you'll have brain cell loss, and basically your mental health would deteriorate. So how does this happen? The normal recessive allele has 10 to 35 repeats for CAG amino acid. But there would be a mutation and you'll have more than 36 genes. When you have more than 36, then you'll get Huntington's disease. And remember, CAG, this amino acid, codes for glutamine. When this mutation happens in the CA gene, then the protein, right, the protein that this glutamine will be misfolded, and that will cause Huntington disease. And remember, Huntington disease can only be inherited from the father and not from the mother. So cystic fibrosis is the final disease. It's an inherited genetic disease. It's caused by a faulty autosomal recessive allele of the CFTR gene. So so the normal CFTR protein, so this CFTR gene codes for the CFTR protein, and this protein is a transmembrane protein at the cell surface membrane. So it's this CFTR protein is a transport protein. And you don't need to know a lot of detail, all of this, but when there is a mutation in the CFTR protein, this transport protein would not be present. It would be faulty. So what happens is this transport protein acts as a chloride channel, and it has binding site for ATP. So chlorine can move out of cell via active transport. Chlorine ions can move out of this cell through this transport protein, through the CFTR protein. And when this happens, right, when you move the ions out, the concentration of water ions would increase. Water concentration increases as chloride ions move out of the cell. So more water would be present inside the cell. So when more water is present inside the cell, then the water concentration increases and you can move more water out of the cell. Yeah, because osmosis happens from high concentration of water inside the cell. So water will move out to the lower concentration. So water moves out of the cell. Why am I explaining this? Because this CFTR gene is present in mucus for the cells that make mucus. So when this protein is not present, when it's absent, then you'll have a faulty protein. This means chloride ions cannot move out. When chloride ions don't move out, then the concentration of water ions would decrease because you have more ions of present inside the cell. So water concentration decreases. So instead of water moving out, water will move in. And what this causes is mucus, right? Your mucus must be dilute. Mucus must be mixed with water at all times so that mucus can move freely on the cell lining. But when you don't have water, when water is absent, mucus would be very thick. And when mucus is thick, it can't move freely by cilia. So mucus would accumulate and it would accumulate, and this causes mucus to trap more bacteria. When mucus traps more bacteria, you'll have more infections. And this would also reduce gas exchange due to longer diffusion pathway. Basically, the mucus would accumulate on the lining of cells. This increases the distance between diffusion. So more distance will mean that oxygen will diffuse slowly into cells. So you'll have reduced gas exchange. Yeah. So this is the symptoms for cystic fibrosis. And so this was everything we learned till now. In the next class, we'll focus on gene expression and gene regulation, how genes are expressed and how genes are regulated, basically transcription, transcription, and translation. All right, so see you tomorrow. Bye. Is everything clear till now? You understood all of this, right? All right. Bye.